Dear visitors, family, friends, and colleagues, I welcome you warmly to the inaugural lecture of Professor Alard Roebroeck. My name is André Aleman. I'm the Dean of the Faculty of Psychology and Neuroscience, and today acting Pro-Rector. An inaugural lecture, or oration, is a public academic ceremony focusing on a newly appointed professor, even though it's already a couple of years ago that Professor Roebroeck was appointed. Alard Roebroeck received his master's and also his PhD degree, cum laude, here at the University of Maastricht. And he has worked as a staff scientist and lecturer at this university since 2006 and as an associate professor since 2014. He was a visiting scientist at Aalto University in Helsinki, in Finland, and at the Research Center in Jülich. Since 2018, he has been head of the Multiscale Imaging and Brain Connectivity section at the Department of Cognitive Neuroscience here at the Faculty of Psychology and Neuroscience in Maastricht. Professor Roebroeck pioneered the international research on brain connectivity, and I think he was the first, together with Elia Formisano and Rainer Goebel, to apply Granger causality in brain research, which was a very important development. He currently pushes frontiers using innovative methods in elucidating brain structure using high-field imaging. Apart from research, he has a strong track record in teaching students cognitive neuroscience, and the Faculty of Psychology and Neuroscience welcomes his appointment as a professor, and we are proving ourselves lucky to have him in our faculty. I could say much more about his work, but I prefer to let the new professor speak for himself. And afterwards, Professor Goebel will give a short Laudatio. But now, the orator himself, may I invite you, Professor Roebroeck, to deliver your lecture. The floor is yours.
Thank you, Pro-Rector. Dear family, friends, colleagues, it's my great honor to give my oration today, entitled Making Connections in Brain Science. And as you may imagine, the main subject of my work and of my talk today is the human brain, a wonderful organ that allows us to do many, many special things that arguably many other animals cannot do, such as give a lecture, or sit in the hall and thinking, oh, is he going to give it in English? That's going to be a long lecture then. All of that our brain allows us to do. So it's a small wonder that it can do what it does. And why is that? Well, as many of you may know, the brain consists of many, many neurons, very special cells. I mean, all of our organs consist of cells, but neurons are very special cells that have a nucleus and a cell body, as all cells do, but they also have many extrusions called dendrites and axons, which can make connections with other cells. We have many of those neurons in our brain, about 100 billion, 100 miljard in het Nederlands. And that in itself, of course, is a very large number, but it doesn't make the brain special, because there are many other organs that we have that have an equal or larger number of cells. Your upper leg muscle has more, for instance. But what neurons do best, as I just implied, is actually to make connections, because synapses, the connections between the dendrites and the terminal buttons of the axons, we have many, many, many more of than we even have neurons. In fact, on average, we have 100,000 connections per neuron, and that is the actual amazing number. No other cells in the body make that many connections with other cells, and that is exactly what gives our human brain the capabilities that it does, and hence we are making connections in brain science. We also have other cells in the brain, by the way. We have glia cells. They were often called helper cells in the past, but we're finding out that they actually do many, many useful things that are important for the function in the brain, such as, for instance, create that myelin that you see over here around the axon, which makes it conduct its nerve impulses much faster than it would without that myelin sheet. And another amazing thing about our brain is the energy it consumes. Not so much perhaps how much it is, because it is only 2% of our body mass, the brain, but it consumes up to 20% of our energy, so it's our most energy-hungry organ in the entire body. But if you compare it to the amount of power, namely 20 watts, that other smart things nowadays, AI, deep learning, ChatGPT use, it is actually a quite amazingly low number. Because by comparison, a graphics processing unit that creates the beautiful graphics in your brain, but also powers all of the chatbots that nowadays you ask all of your questions and give you back wonderful answers. Well, just one GPU will actually use 100 to 200 watts of energy. And then all of these wonderful new AI technologies, and I'm not talking down on them, I think they're beautiful. I think they're going to be able to do even much more, even do things that we thought only we could do. But however good they are going to get at it, they use astronomical amounts of power. So much, in fact, that I just took this from a couple of news websites that talk about how big so-called hyperscalers, those are the Microsofts and the OpenAIs that build these chatbots, now actually are looking to put down more nuclear plants in order to power all of the GPUs, so many of those power-hungry GPUs, to power all of those chatbots. And so you start to wonder, how amazing is it that the human brain can then do that at only 100 watts? And that has been the topic of my research, of my career, and of the research of my group, about which I want to tell you a few things today. Part of the secret of why our brain, the human brain, is so amazing at doing these complex things is the cortex, the neocortex. We also call it the gray matter. It's this wrinkly outer sheet that you often see in pictures of the brain. And if we make a cut through it and look at it from the inside, you see that it is really a sheet. It's very thin, and it's on top of what we call the white matter, and we see why we call it the gray matter. By the way, it's only gray when the brain is dead, sadly. A live brain is actually not as gray. And then, let me try and advance my slide, which at the moment it doesn't want to do. It seems that PowerPoint was a bit slow. Another computer that is very power hungry and is not as good at things as the brain is. This thin sheet of gray matter that we have up here that does most of the work for the most difficult things that we're capable of doing is about 2 to 4 millimeters thick. And it consists of many, many, many cells, which you see over here. And when you look at the size and the division of those cells, we call that the cytoarchitecture, cyto for cells. All of that myelin along those axons also has a very special pattern. We call that the myeloarchitecture. And each of those you can use to actually divide the neocortex into 6 specific layers. The neocortex has 6 layers all over the brain. And in those layers live cortical circuits, local circuits of connections that the neurons make with each other that perform these capacities that we have. But there are not only local circuits, which are, by the way, quite different between different areas of the brain. The auditory areas have slightly different circuits and layers as the visual areas or the prefrontal areas or the parietal areas. But also they are very widely connected through the white matter and across the brain. So really, the human brain, the human neocortex, is a big collection of small cortical circuits that are connected remotely to other cortical circuits. And it's all those connections together that make it do what it can do. Well, that, you may say, is quite complex. Yes, it is. And so how to study that? Well, that is a very difficult task because in all of those cells, with all of those connections, you have to consider that you're working across many, many scales of the brain. You can measure things about the brains in centimeters. You can look at the entire brain. You can measure it in millimeters. You can measure it in micrometers, such as the individual neurons or perhaps the layers that I talked about before. You can even measure things in nanometers, like those synapses, those individual synapses that make the connections. So how can you measure all of that? Well, I'm afraid that you can't measure all of that all at the same time. We have to use different techniques, such as magnetic resonance imaging, MRI, light microscopy, LM, and electron microscopy, EM, to study different parts of the scale continuum that you see over here. And each of them will have a different typical resolution, maybe millimeters, and a typical field of view, maybe centimeters. And electron microscopy, you can look at nanometers, but you can only do this over maybe 20 micrometers. And so the trade-off between the resolution you can look at, how small your pixels are, and how much you can look at, your field of view, is always a difficult one. And to play with that trade-off and to make measurements of the connections in the brain with different techniques and combining them has been what in my group we have done for, well, about 20 years now. Today I will talk about ex vivo MRI, applying magnetic resonance to the ex vivo brain, the dead brain, the cadaver brain, ex vivo light microscopy, and of course then in vivo MRI, doing MRI on the living, working brain. MRI, magnetic resonance imaging, what is that? Well, a brief introduction. It's this big machine that you may have seen in the hospital once, that has a field strength that we measure in Tesla, T. It's not the car Tesla, it's actually the unit of magnetic field. And so a 3T has 3 Tesla of magnetic field, makes wonderful pictures of the brain that is also used in the hospital. But a 3 Tesla is a clinical machine that is nowadays surpassed by research machines such as a 7 Tesla MRI scanner, and even here in Maastricht, quite unique, a 9.4 Tesla MRI scanner. So more and more Teslas, bigger machines, also more expensive machines, which makes you ask, so why? Why these bigger magnets and why more Teslas? Well, because it allows us to measure more contrasts, because with the MRI, quite uniquely, you can measure different aspects of the brain. You can have, you could say, different image filters that allow you to wait for different entities in the brain, like myelin, or cells, or iron. Here you see T1, T2 star, and proton density contrasts. You can measure more of those. You can measure higher resolutions. That's intuitive. I already made a point that that might be important. You can cover more of the brain. You cannot just measure a part of the brain. You can actually measure the whole brain in a shorter measurement time. Of course, all these things interact. Even with a 7 Tesla or a 9.4 Tesla, you cannot get all of those things all of the time, but you have more choices to make. You can measure more of the brain in shorter time. And that's needed, because when you want to study this really thin sheet of 2 to 4 millimeters of cortex, then your voxels, which are volume elements, our pictures on our phones, our picture elements, pixels. In our imaging apparatus, we have voxels because they're all volume elements. We call them voxels. You will hear me use that term a lot. Our voxels, traditionally on a hospital 1.5T or 3T MRI, are 1 to 2 millimeters. And you can already see that that is not enough to actually look at the layers of the cortex. We will actually have to make our resolution higher. We have to make our voxels smaller in order to even just sample the different layers of the cortex. And that's what we've done over the last 10 years, ex vivo, with the 9.4 Tesla and the 7 Tesla, where we've also built specialized hardware, so-called radio frequency coils, which is what your head is in or what the sample is in when you scan it. And if you innovate those more, you can also get even more resolution out of your scans. And then, since they are cadaver brains, scanning them over an entire weekend, and afterwards crunching many terabytes of data in order to reconstruct images of the brain. But then we can get images of the brain at resolutions that are about as high as the thickness of a human hair. So down to 50 to 120 microns. And here is a section, and actually if you see the brain here, it is this section, a so-called coronal section through the back of the brain. Coronal is like this, like a corona would be over your head. Here you see a measurement that we've performed in this way that has a resolution of 100 micron. And then just zooming into this part over here, this sulcus, this fold of the cortex, is the so-called visual cortex, where the signals from the eyes go all the way to the back of the brain and where they are first processed in the brain. And the visual cortex has already known for a couple of 100 years under a real microscope, an optical microscope, to have a specific signature, which is called the stripe of Gennari. Of course, it was a doctor called Gennari that first saw it. And that is only visible, or was only visible, under a microscope of very thinly sliced brain preparations. But now you see that when we can actually keep the brain intact and measure at 100 micron resolution, we can actually see the stripe of Gennari running along the cortical ribbon and see that V1 starts about here and ends about here. And so in this real sense, we have the potential to create an in vivo microscope and look at the structure of the brain. Here's another 100 micron depiction of a brain that we used. you may notice that one hemisphere, one side of the brain, looks a bit different than the other hemisphere of the brain. If you stare closely, and I hope the projection is good enough, you may see some differences. And that's true. This is actually not one brain. It's two halves of two brain cut in half, where this is a relatively healthy hemisphere. Well, the person is dead, so of course, that is in a relative sense. At least there was no known neurological deficit, but this is a person that died of Alzheimer's. And indeed, you can see that Alzheimer's, as is well known, starts, and I'll zoom in a little bit, with pathology in the so-called hippocampus, which is this structure over here, which later on actually spreads over the entire cortex, and later on is also then often coincides with vascular defects. And so at this resolution, we can actually see these defects. Now, of course, ex vivo, but the hope is that we can later do this in vivo and detect it early. This is the highest resolution ex vivo scan that we ever did, 75 microns. So where 100 micron is already 1,000 times higher resolution than 1 millimeter isotropic, 75 microns is actually 2,000 times higher. And now, zooming into this part of the brain over here, I hope that the projection might be good enough for you to see, especially if I point it out, that not only can we cover the entire human brain and tell what the white matter is, which, by the way, here now is black or gray, and what the gray matter is, which now here looks white, so that's confusing, but you have to take it from me. But you can also now start to see signs of the myeloarchitectonics of the brain, once again in an intact brain, whereas 100 or 200 years ago, we could only do this by slicing the brain very thin. Another way in which MRI can become an in vivo microscope. Now, the topic, you may ask, Professor Roebroeck, of your lecture was connectivity. So where is connectivity? Well, we can also do that with MRI, with a very specific technique called diffusion MRI. I'll tell you a little bit more about how it works. It works with the diffusion of water, but it actually allows us to look at the white matter, right, which again, remember, looks gray now. And you can see that we can actually trace the local directions of the long axons that run through the white matter. We can do this not quite at 100 micron, but we can do it at 400 micron, because it's a bit harder to do than the previous results that I showed you. And that then, at that resolution, allows us to look at how these long-running connections run from one circuit to the other, and then actually insert into the gray matter, which they do with very special patterns, which are actually very hard to produce, especially when you have low resolutions, like 1 or 2 millimeter, but at 400 micron, we could reproduce these much better. That brings me to trying to go at even better resolution, at cellular resolution. We have not managed to do that, not precisely, but I'm coming back to that point, with MRI. But we have been able to do it with light microscopy, which has a bit of a different field of view versus resolution trade-off. But we have to say that when we have now managed to make our imaging voxels, roughly, let's say, 100 to 300 microns with MRI, about 10 years ago when we started that work, that was the entire field of view, 100 to 300 microns, of what a microscope could do. It could look very finely at cells, but only in a very, very, very small square. So there, we had to try and extend the field of view, keep the resolution, but extend the field of view. And the quintessential technique that came along that we started using was so-called light sheet microscopy, where you actually use a laser to shine a sheet of light, very thin, micrometers thin, through your sample, and then, while you move that sheet through your sample, you make pictures through a microscope objective in order to get very, very high resolution, and importantly, 3D pictures of the individual neurons in the brain. And now, much more than microscopes before, which could already do something like this, confocal microscopes and two-photon microscopes, you can do this very quickly for a lot of field of view for very large pieces of tissue. And you can see here how you can see all of the individual neurons. You can clearly see, I hope, sorry about that there's a layer transition here that we're running through. This is actually layer 1 of the cortex. This is layer 2 of the cortex. And by now you can't see that, but we're actually several millimeters into the tissue. How, you might ask, does that actually work? Because, I mean, can you still capture the light when the brain is very opaque like that? Well, no, you can't. You need to do one additional thing, which is called tissue clearing, making tissue transparent, making tissue see-through. That's a very difficult process, and that together with light sheet microscopy allows us this power of looking at cells over very large pieces of tissue. The tissue clearing is a chemical process pioneered in our group by Sven Hildebrandt, from whom I will show a bit more work later, that actually allows to perform light sheet microscopy into human cortical tissue. And with that, we have actually been able to establish what for me, at least 10 years ago, was a bit of a dream, namely to go from the brain to a neuron, to the cell, to go across all scales. Not with one technique, but at least with two. Here you see MRI. This is actually, once again, the visual cortex, that same solstice over here, the calcarine solstice, where the visual cortex is here again, but we're now looking at it from the back. And we performed that high-resolution 200 micron MRI on it, but afterwards, we actually took these two pieces out and then performed light sheet microscopy on it. So here, you have the front piece, where we made it transparent, we cleared it, performed light sheet microscopy, and can already see the layers of the primary V1 and the secondary V2 human visual cortex. And then zoom in further, and now the solstice has actually been inverted. It looks downward in the middle, and it looks upward in the bottom picture. And actually look at the cytoarchitectonics and define all of the layers of the human cortex in that way. If we look at that other piece, the front piece over here that's indicated here, again, we were able to make that transparent. This time we focused on V2, the secondary cortex, and again, we're able to perform all of the layering and the cytoarchitectonics. And mind this square over here, we'll try to magnify that a bit after I've shown that this here is not a 2D slice, as we used to use, but a 3D data set, which once again shows all of the individual cells and all of the layers of the cortex, and does that over that entire millimeters thick piece of cortex that we've made transparent. And by now also, I will leave it running, you will see that we can go many, many millimeters into the cortex and see its cells and see its cortical layers. If we then zoom in further, we can actually see that the resolution is good enough for us to actually find and reconstruct single neurons in 3 dimensions and know exactly where they were in that MRI scan that we made earlier. So that's our best attempt so far of going from brain to neuron, from brain to cell. But we have not stopped there. We've continued innovating, and one of the nicest innovations that we've performed, which involved both software and hardware, was building our own light sheet microscope, the so-called CT DSPIM, the cleared tissue double selective plane illumination microscope. You don't have to remember that term. Let's just call it a CT DSPIM. That's a difficult enough abbreviation. The idea of the CT DSPIM microscope is that if you take a piece of tissue and you shine your light sheet through the side of it and look from the bottom, the piece of tissue cannot be very large because your sheet will not penetrate anymore. So what if you put your microscope at an angle from the top and then shine your light sheet in at 45 degrees and also measure, make the pictures through the objective at 45 degrees, then you are now capable of moving your tissue underneath your microscope and laterally measure very, very, very large pieces of tissue, of which the data will look like this. Of course, it is now a weird way of going through your data because you're moving sideways and you're moving in depth. But you can correct for this when you create your 3D images. And then when you do, and that's our latest work, we can now make reconstructions at cellular resolution of very, very, very large pieces of human brain. And see the layers, as indicated here by the arrows. Once again, you see there a transition between primary and secondary visual cortex, but also go all the way from layers via cortical columns, another structure that is very well known in the brain, to single cells, and in 3-dimensional data sets, start counting these, the many thousands of them, for the individual layers of the cortex. And in that way, get closer and closer to that mesmerizingly complex architecture of the brain that we want to learn more about. Now, all of that, of course, was performed on ex vivo tissue samples. Of course, the brain does what it does when you're alive, and you would love to see a functioning brain. And that is why, in the last part of this talk, I will focus on the work that we did with in vivo MRI. And at first, this is focused on the structure of the living brain and how the connections change by learning, by getting older, both getting older from being 8 to being 14 and getting older from being 50 to 80 years old, because structure of the brain does change all of the while, and that is actually how we learn. Well, there comes that technique again of diffusion MRI that I referred to before when I showed you the directions of the white matter tracts. Diffusion MRI is actually an MRI technique that can non-invasively, so you lie in the magnet, it doesn't do anything harmful to you, it doesn't actually even use radioactive substances, it can non-invasively measure the diffusion of water, which means the thermal motion of H2O molecules. Now, of course, not the individual molecules. Every single voxel will have many trillions of molecules, but the scanner can then see the average motion of those molecules. They will be moving. They will have Brownian motion, the motion that you see when you put a drop of ink in a glass of water. It's that same motion that makes individual molecules move away from where they were with time. Now, when they do that in a glass of water, of course, they will just move further and further away. But when they do this in the brain, when there are dendrites around or axons around, they will bounce off those barriers, and that, of course, will lead to motions, diffusion, that is hindered and restricted by those barriers. So, for instance, here, the water molecules will move much more in this direction in between these schematic axons that I drew here than they will in this direction, because they will bounce more often. And so by the way that many of these water molecules bounce off the tissue, we actually know more about what kind of tissue lives inside our voxels, however small or big they are. For instance, let us make a drawing of gray matter, right, where that neuron with its soma is, and white matter, where many of the axons with the myelin are. And consider that a voxel that we measure here, that little cube that could be 1 by 1 by 1 millimeter, it could also be 100 micron cubed, consists of different type of compartments. It has water that is inside the axons, inside the tubes. It has water that is outside of the axons, outside of the tubes. And of course, there can also be glia cells, like the ones, the oligodendrocytes that make the myelin, that are roughly spherical, inside of which the water could be bouncing around. All of that water behaves differently while it bounces around. And so we can then take our voxel signal and see what is typical for intra-axonal water and what is typical for extra-axonal water. The intra-axonal water will bounce around differently. It will stay closer to where it was than the extra-axonal water because it can actually escape at a certain point. And so we can tease that apart and then inside the voxel create maps of things that are microscopic, such as the axon densities. How many axons are there inside of this voxel? And, as before, we can also see the axon orientations. We can see that there's probably a large bundle of axons running in this way and one running in this way. And by the way, you might say, it looks like everything is crossing everything. Yes, that's actually true. In the brain, there is no nice roundabouts where axons wait for each other. They actually do cross all over the place, and you have to be able to reconstruct that with your imaging. It's a little bit slow again, by the way. That gives me a little bit of time to maybe explain. Now it's very fast. There we go. That's where I wanted to be. Apologies. We've been able to take that a little bit further, especially with our 7 Tesla magnet that can give us more contrast and more resolution, and actually also look at not just axonal density, which I just explained, but also axonal diameters, at least average axonal diameters, how thick the tubes are. In this case, that is actually over the so-called corpus callosum, which is, if we take the brain and we now make a so-called sagittal cut through the brain, that's this dimension here, we will actually see that in the middle, this big bar that connects the two hemispheres is the corpus callosum, and over that there is an interesting variations of axonal density and of axonal diameters. The large diameters actually connect the motor areas, and the small diameters actually connect the frontal and the visual areas at the back. And that has actually specific things to do with how these areas function, because you can imagine for motor actions, you need to be fast, and therefore the axons are thick. And we have our sensitivity for this. We also have sensitivity for the myelination of those tracts by applying yet other techniques. And so we arrive at a future that I want to sketch for you, where we are using and are going to continue use and are going to continue to push forward MRI as a so-called in vivo microscope. With that, of course, I mean that we want to make our voxels smaller, but also especially that we want to see the average cellular microstructure inside those voxels, especially with diffusion MRI. What axonal diameters live inside this voxel? How many axons live inside these voxels? How large are the cells inside these voxels? And for that, we need MRI measurements, which are fast, which acquire many contrasts, such as you see here, diffusion contrast, T1 contrast, T2 contrast, and they do this over the entire brain. One of the sequences that we pioneered is the so-called mesmerized sequence. It's not important to understand exactly what this means, but this is a pulse sequence with which you control the scanner, with which you indeed can acquire many contrasts over the entire brain very fast. And so we are trying to pioneer the MRI scanner software that allows us to do that. Then the data that comes from that need to go into microstructure models, computer models, statistical models, which actually tell you from your measurements what the axonal density might be, or the axonal diameters, or the cell sizes. And those need to be robust and reliable models in order to be an in vivo microscope. And of course, one very important step that we are undertaking and that we are continuing is to validate these ex vivo with ex vivo microscope, such as the light sheet microscope. And that has many applications, not just fundamental brain research, understanding more of how our brain does what it does, but also especially in disease to see what goes wrong, such as the early detection of Alzheimer's disease, Parkinson's disease, and multiple sclerosis. Also, a particular point of focus for us very recently is to try and detect and delineate focal epileptic lesions that are often undetected on clinical 3T MRI exams. The future then for light sheet microscopy, and particularly the CT DSPIM as an ex vivo microscope. Well, there we are really pushing forward, and particularly Sven is doing that, in performing 3D histology, so to look at tissue at cellular resolution in 3D, such as I've been showing you, and to have faster measurements to look at more tissue in 3D and at higher resolutions. And especially also, of course, we can only look with those fluorescence laser microscope at things that we actually label, where we can attach small molecular labels, and we need more kinds of labels, such as, very recently, for instance, we found a label for brain vasculature. Here you see the brain vasculature of the primary and the secondary human visual areas over the depth of the cortex. That is very important, for instance, for something like stroke, but it's also very important for another technique that we ourselves, and even more my own colleagues use, functional MRI, because that is all about how blood flows through the microvasculature. And then another application is, of course, in the same vein, 3D pathology. So to do 3D examinations of medical resections and punctures that were taken from people that have a suspected disease or cancer, and then to do en bloc, so entire intact tissue pathology when, of course, we clear those samples, when we optically clear those samples. We've already performed this in our most recent work for prostate cancer, and here indeed you see some CT DSPIM light sheet images that we've performed for prostate cancer. That brings me to my acknowledgments. In becoming a professor, there are many, many, many people that you could think, and I have to start with saying that it's impossible to thank everyone. Now, that might sound as something that everyone says when they do an acknowledgement, but it's really true when you actually prepare an acknowledgement. And yet I'm going to try to thank people that were essential in creating, in supporting my career and putting me where I am now. First, there's Rainer Goebel, my mentor since very long. I came to you a long time ago as a student, and I think it's entirely to your credit that I never left. Even time I felt like I sometimes tried, but you've always managed to enthusiasm me in such ways that I found it more convincing to stay. Thank you for your friendship, for your guidance, and thank you for all of the good advice that you've given me, such as always follow the research plans that are closest to your heart. Ilya, another member of my, where are you? There, over there. My co-promoter. I know you equally long as I know Rainer. Also you, thank you for the friendship and the guidance. And thank you especially also for pointing out that you can always follow, of course, your best ideas, and you should, but there are many practicalities in actually getting it funded and getting it done. Sonia, where is Sonia? There you are, of course. Another member of my supervisory committee. Thank you so much for your guidance, for your friendship, and also for those wonderful vegan meals that you sent us in a difficult time. And sorry to shock you at a certain point by telling you that I'm actually not a vegetarian, but that we eat vegan at home. Peter de Weert. Dit kan ik in het Nederlands doen. Ja, je bent een heleboel. Je bent een lid van de begeleidingscommissie. Je bent head of department. Maar je bent ook een vriend, een collega. En zeer bedankt voor al je hulp, voor al je advies, voor al je vriendschap. Zeker ook in wat een erg moeilijke periode was. En niet alleen namens mij, maar namens de hele faculteit en vakgroep bedankt voor jouw efforts in het bewerkstelligen van de Bachelor Brain Science die we nu hebben, waarin we dit soort dingen kunnen gaan onderwijzen aan studenten die misschien de nieuwe generatie kunnen zijn van Brain Science professors. Then, my group, my lab, the CBC lab, the Computational Brain Connectivity Lab. Many have come into the group and have gone out again, and I've decided that what time I had was too little to thank them all. But at least I can say, everyone who was part of the group, thank you for your contributions and for your wonderful work in our publications. Two people at least I want to spend a little more time on. First, Sven Hildebrand. Thank you for your unstoppable enthusiasm for brain anatomy, histology, and microscopy. Your stamina and enthusiasm is just unbelievable. I mean, building microscopes and a wet lab in your own kitchen just is the beginning of how passionate you are about creating techniques and why much of what I've shown today was actually due to your work. Second, Michael. En weer in het Nederlands dan. Ik zeg vaak tegen mensen dat je mijn rechterhand bent. En dat is nog niet eens de hele waarheid. Waarschijnlijk ben je mijn rechterhand en mijn rechterbeen. Zeker als we gaan mountainbiken trouwens. Wat we misschien wel wat vaker moeten doen, maar dan moeten we wel niet zo vaak vallen. Want dat zorgt er meestal voor dat we ons rechterhand niet meer zo goed kunnen gebruiken. Bedankt voor je vriendschap, voor je steun in deze laatste jaren en voor het maken van CBC Lab tot wat het is. Ben, where's Ben? There's Ben. Thank you for collaborations, for friendships, collaborations on pulse sequences, some of the ones I've mentioned we've done together, and for conversations about all things in and out of science. I thoroughly enjoyed those. Federico, who is not here at the moment, and he's told me why, because he was traveling, but he gets to be there at the party. I'm not from the south of Italy, Federico is, but when I have discussions with Federico, some Italian passion sometimes takes over and our discussions rise in volume. That just says how much of a friend and how much of a sounding board Federico has been for me in late years. Milene. There's Milene. Weer in het Nederlands. Bedankt voor je vriendschap aan mij, aan Amanda, voor vele jaren al. Voor je advies, voor je hulp en ook voor het koken met de kinderen, wat ik altijd erg leuk vind en wat ik hoop dat we gaan blijven doen. Vincent. Ook bedankt voor je vriendschap, voor je advies, voor je gesprekken, voor je steun en ook voor het gitaarspelen natuurlijk. Wat ik hoop dat we weer meer gaan doen de komende tijd, als het minder druk wordt. Alle andere collega's in de groep en daarbuiten, bedankt voor de steun, voor stimulerende discussies en ook voor velen van jullie, net zoals Sonja, voor de heerlijke gerechten die er maandenlang zijn gebracht op dinsdagavonden toen ons gezin het erg moeilijk had. Ten slotte wil ik, wat werk betreft, niet stoppen zonder ook Bandito te bedanken en het team van Sejan en Quentin en Diane en alle andere core teamgenoten zoals Jana, Julie en Emma voor vele jaren van heel erg goede koffie, want geloof me, zonder die hoeveelheden koffie was ik nooit professor geworden. Op een persoonlijke noot. Jolanda. Om het op het werk goed te laten lopen, moet het eerst thuis goed gaan. En jij bent al die jaren onze steun en toeverlaat al thuis. En de laatste jaren nog maar veel meer. Je bent echt deel geworden van ons gezin. En bedankt daarvoor. Tante Lian. We delen een verjaardag. En we delen ook de zorgen voor het opgeruimd houden van het huis. Dat is nogal een uitdaging. En zonder u zou ik het echt niet kunnen. Bedankt dat u al die lange jaren al zo'n steun en toeverlaat bent geweest. Zus, broer, Laura, Arthur. Bedankt voor de onvoorwaardelijke steun en liefde. Ook al is het vaak op afstand en spreken we elkaar niet zo vaak als dat we misschien zouden willen. Het is er en het is er altijd. Gelukkig vinden we tijd voor vakanties in de bergen samen. En ik hoop dat we dat blijven doen. Nico, Annelies. Bedankt dat jullie de beste schoonouders en de beste opa en oma voor Maarten, Jonathan en Tanne zijn die ik me maar kan voorstellen. En bedankt voor al jullie steun en liefde over al die jaren. Pap, mam, tja, wat moet ik zeggen? Zonder jullie was ik er überhaupt niet geweest natuurlijk. Maar zonder jullie opvoeding was ik ook niet geworden wie ik ben. En daarvoor dank. Heel veel dank. Pap, van jou heb ik geleerd dat het waard is om wat je doet goed te doen. En je in de details te verdiepen. En dat passie voor je vak mooi is en goed is, ook al is dat niet makkelijk als je vak druk is. Mam, bedankt voor het ondersteunen van bijzondere ideeën die ik altijd gehad heb, als kind al. Ik noem een herinnering een sportschool bouwen in de garage van hout. Of in de eind jaren 80 software zoeken die muziek kan opnemen, wat toen heel hoogst ongebruikelijk was. Jij hebt dat altijd ondersteund. En je hebt me ook geleerd onversaagd te durven iets anders te doen. Vooral als dat is wat je voelt dat je echt wil doen. Maarten, Jonathan en Tanne. Van alle dingen die ik in het leven heb, ben ik het gelukkigst en het blijst met jullie. Maarten, wat ben jij goed in de ballen in de lucht houden. Zowel letterlijk als figuurlijk. Hè? Letterlijk met voetballen, want daar ben je heel goed in. Maar vooral ook met alle dingen die je doet. Wat doe je dat knap. Je hebt je zaken voor elkaar. Jonathan, wat een geweldige focus en inzet heb jij voor dingen die je wilt en die je wilt doen. Als jij een VR-bril wilt en ik zeg dat je niet genoeg spaargeld hebt, dan is dat voor jou geen probleem. Je vindt toch wel een manier. En zo doe je het ook met de dingen die je wil leren. Heel knap. Tanne, wat een mooi invoelend vermogen heb jij. Wat kan jij goed zien en voelen wat mensen denken. En wat leef je dan mooi mee. En wat ben je creatief en wat kun je mooi tekenen en zingen. En dan, tenslotte, de reden dat het voor mij heel lang heeft geduurd dat ik hier stond, is dat ik me niet voor kan stellen dat ik hier zou staan en Amanda niet zou kunnen danken. Dat kan ik nog steeds niet. En dus doe ik het maar zo. Amanda, jij hebt me geleerd dat het leven gaat om mensen. En om te communiceren met mensen en het dicht bij je houden van mensen. En ik ben je daar heel dankbaar voor. Ook al moet ik nu je wijze woorden en je aanwezigheid missen. Dan moeten wij dat missen. En ook in de wetenschap weet ik dat me dat een betere wetenschapper heeft gemaakt. Want ook daar weet ik door jou dat het niet alleen gaat om de programma's die je schrijft en de paragrafen die je schrijft. Maar om het samenwerken met andere mensen en om die dicht bij je te houden en te begrijpen wat zij willen. Het was moeilijk en het gaat moeilijk blijven om het zonder jou te doen. Maar we proberen het en het gaat goed. Ik heb gezegd.
Alard, I'm very touched. Thanks so much for this wonderful lecture. And I have to say that I hope I have helped a bit to guide you through your life professionally at least. But you also did it for me. So I really can say that, I appreciate you as a person, of course, also as a brilliant scientist, which I try to show in a moment, very much. And I want to say that for me, but also especially also for Elia, we together, we really enjoyed having seen you growing up in science, from a master student, bachelor master student, all the way to a full professor. And it was really joy giving one episode of your life, not of course. So what I want to say now is, I want to do this Laudatio very personal, just give some memories I share with you, and with some others here, of course, but it's really very personal. And the first thing where you basically, popped out, so to say, was when I arrived here in 2000, January 2000. And I think one of the first students I talked to was Alard Roebroeck. You were there already, and that means that in January, next January, we know each other 25 years. At that moment, when you came to me, you were very determined. You came to me, oh, Professor, do you support me studying both psychology and computer science? It's not easy to get this done formally. There are all these hurdles. And then you saw my face. I smiled. And I told you, I do everything I can as this new professor because I studied both myself. And I was so happy to found a common soul here, right? So the first student said, wow, you know, all the people tell you when you get other application talks, we have the best students. And then you came, right? And it was, for me, really important to feel happy here in Maastricht. So when I had this feeling, this is a very special student, at some point when we started to talk, I wanted to invite you to our place. And you came, and that's where I really realized this guy is really special, because I showed you all my nice beverages and say, listen, this is a good whiskey, you should try this one, and over this one here, it's a very good one. And then Claudia at some point said, yeah, but Alard, what can I offer you now? And you said, do you have a glass of milk? And we still smile when we think about you, about this event. It was really touchy for us. Yeah. And of course, having such a bright student, as expected, he finished his bachelor and master, as the pro-rector has alluded to already, of course, with cum laude in 2002. And you started, and Elia and I were really eager to offer you a PhD so that this guy is not disappearing somewhere, right? And I remember very well that a big event for you probably also, for us too, Elia and me, but I think I saw how it impressed you was when we decided to go together to the first connectivity, functional connectivity meeting in Düsseldorf. I think that was so impressive. I saw your eyes open all the time, seeing all these big shots, Carl Fristen, Kötter, many others, right? It was really so impressive, Carl Zillis. And actually, I remember how we discussed a lot Carl Zillis' lecture, because he said at that time, I don't trust this new diffusion MRI thing, because in the brain you have more than 45-degree curvature. And so he made very clear to us that it's probably much more challenging than, at that time at least, the early diffusion-weighted stuff, DTI stuff, right? So therefore, I remember that well. That was quite impressive, I think, also for you. And I know that you already then, although we focused first on something else, but you already then got this idea in your mind, and you did it over many years, namely to compare and validate what we see in real postmortem tissue, whether it's, for example, the kissing and crossing you alluded to, whether we can really reconstruct it using this kind of technology, diffusion MRI, and analysis of that data. So what we then first both shared was the idea to apply this Granger causality idea to fMRI. And you became really a pioneer because you were the first who did that. And that is, for me, was one of the most impressive things to see, that you, more or less alone, I mean, Elia and I, we helped a bit, but more or less alone, you wrote the mass down, you coded the stuff. I've still in my software framework your plug-in on this. and you wrote the papers. So I was impressed how complete you are as a scientist, as a PhD student, right? This is really rare, and you really impressed us. Of course, we learned a lot, maybe more from you at that time than the other way around. But it was really an impressive time to see you grow and see your capabilities. You also realize that you have done something. It's not only that this is one of your seminal findings, but it's one which really spread. So people contacted you. It was another experience for you. And you asked me, Rainer, shall I help these people? How much should I do? You know, because many people approached you and said, can you help us applying Granger causality, right? So that was also, I think, a great experience to see, how this goes. And I remember very well an episode, I think it was after an HBM or at an HBM, when I wanted to go, I think it was in US or Canada, and I wanted to go my usual 2-year Stanford visit. And then the people told me there, oh, you should tell about this Granger causality and this new diffusion stuff and so on. I said, yeah, listen, the guy who did that is with me at HBM. Oh, bring him over. And so we both went together to Stanford, and I have a wonderful picture of that, which you will get later. And I think that also is one of my memories when I thought what to talk about. And you, of course, impressed the people there, so that Brian Vendel basically said, now I'm also doing diffusion imaging, right? He was not so much convinced at that time. So he had also there a huge impact. You don't know this probably, but you can ask him. And then something interesting I wanted to also share, because it shows really your brilliance. There is, of course, not only Granger causality mapping. More or less at the same time, Carl Friston and colleagues developed dynamic causal modeling, right? And it's also cool. So there are 2 different methods. They have their pros and cons, you know. But when a paper from David 2008 came out, Carl wrote a very strong piece where we all nodded our head. What did he do here, right? Because he basically killed Granger causality. He says, since it can be that you get the order of effects across areas, the connectivity, wrong, that means Granger causality should not be used. It's basically not working, right? It was very strong and very harsh. And then you wrote basically a response to that, where you analyzed all the different components that you differentially compare, how to do hemodynamic, that also the tool tells you when not to have a direction effect. And you explained all the things you did already, most of it 2005 in your new image paper, but you repeated it in a much even more in-depth way. And then something happened which I couldn't believe, because it shows also greatness of Carl Friston, I have to say. But in a reply of him in 2011, the same year you published your paper, he wrote a response, just he as the author, and he started this paper. I apologize for the superficial and dismissive treatment of Granger causality. This is impressive. This is based on your paper. And I wanted to share that. That shows what you write, how you write. You know, this is brilliant, and it is logical. This is conceptually deep and so convincing that really great people like Carl Friston acknowledge that. It's a big achievement. And the story was finalized because you, and another friend of us, Pedro Valdes Sosa, you wrote a wonderful piece where you showed that there are actually abstract principles from which you can derive when you make choices, probabilistic, deterministic, and whatever, to end up in the different methods which are around. So just to show, to express the brilliance you have. I actually have a lot of here, but I think I don't want to make it too long. But when I started thinking and writing, lots of thoughts ran through my mind. But I want to not forget to stress what you showed in your talk. You became not only a pioneer in Granger causality mapping in fMRI, but you also became a pioneer in diffusion imaging. What you did there is also what all the people I meet and talk about know and talk about. So it's really another big achievement. And also your recent work you have also shown here, which I'm not so much involved, but I observe it with great pleasure. And it's really remarkable. For me, the most remarkable is how you really bridge the scales from, as you said, from the macroscopic 9.4 Tesla data then and down to the cells now with light sheet microscopy. So it's really impressive. So just to see that you shine now at least the third time in a really pioneering way. You build your own microscope. Who does that, right? And that brings me to another point I just want to mention very briefly, how instrumental you were for setting up both our first scanner, the Allegra, and maintaining it together with us, and we together, but even more so for the high field scanners. Because I was just flattened to see how you converted also Federico into MR physicists. So you negotiated tenders, you knew all the details of all the specs, you know how all the things work, and after we got the stuff, we wrote a grant together to get money for building coils, which you did unsupervised and did yourself to some extent, right? So this also would also show that your skills are not just, in parentheses, you know, mentally, you really are really good also to guide and build things. This is really, I think, an important point. Now I want to come to the end. I just want to say that, yeah, that I also admire, you know, Amanda was in my section and worked very closely with me, and we were all struck by that. But I admire how you managed to go back into your life and also to go back to work. I know how tough that was. We talked a few times about it. And it's great to see you, and I'm really proud of you. And I can only say, this is wonderful that we can celebrate together with you this event. I also want to congratulate your family. Jullie kunnen allemaal trots auf Alard sein. And I want to thank everyone here, friends, colleagues, who share this wonderful day with our brilliant scientist and wonderful person, Alard Roebroeck.
Thank you, Professor Roebroeck, for this fascinating oration that you gave, which marks the inauguration of your chair. I hereby close the official part of this academic ceremony. A few household matters. We are about to leave this lecture hall, where the cortege will go first, and my advice is not to stand behind the professors in the corridor, but to walk straight to the refectory and to keep an eye on the orator and to find a suitable time to congratulate him. Thank you very much.